Hello, everyone, and welcome. We want to thank you for joining us for today's webinar. Before we get started and while people trickle in, I want to just introduce Nanoimaging Services and introduce our speakers. Nanoimaging Services is a US-based cryo-EM service provider focused on structural biology and nanoparticle characterization services. In January of this year, we acquired Proteos, a protein production CRO based in Kalamazoo, Michigan, so that we now offer the full gene-to-structure workflow as well as standalone protein production. So today, we're going to be focusing on a protein degrader complex, and we'll showcase our full gene-to-structure workflow using Cereblon DDB1. We'll be walking you through our process of producing high-quality structure-grade Cereblon DDB1, as well as our Cryo-EM workflow for solving the structure of the complex, both in the apo form and bound to ligand. During the webinar, we encourage you to post your questions in the Q&A toolbar, and then following the talk, we will answer your questions live during our Q&A session. Now, without further ado, it is my pleasure to introduce today's speaker, Dr. Giovanna Scapin, who is joined by Dr. Lindsey Gottler. So Giovanna is the CSO at Nanoimaging Services. She joined our team after gaining extensive industry experience in the field of protein structure and structure-guided drug discovery during her time at Merck. Giovanna is an accomplished crystallographer and learned Cryo-EM from the New York Structural Biology Center so that she could help establish the Cryo-EM facility at Merck and promote the use of Cryo-EM for drug discovery. Today, Giovanna will be joined by Dr. Lindsey Gottler, who is the VP of Scientific Operations at Proteos. With over 20 years of experience, Lindsey has deep expertise in recombinant protein expression and purification. She specializes in optimizing expression conditions across E. coli, insect, and mammalian systems, and designing purification strategies for diverse targets such as enzymes, antibodies, and multi-subunit protein complexes. Please join me in welcoming Giovanna and Lindsey. So Giovanna, I will pass it over to you.
Thank you, Sarah, for the very nice introduction. And thank you all for attending the webinar. So let's get started. I will start out with something that I have not shown in quite some time, which is the fact that samples that are submitted for 3D reconstructions are not all the same. But each one of them has some characteristics or peculiarity that makes it different. And over the year, we have grouped these characteristics into buckets. Now, the size of the bucket may need some updating right now, but the message is basically still the same. Only a small fraction of the samples that we receive are actually good samples, meaning that allow us to go from sample in a tube to a well-resolved sub-3 angstrom map in a couple of weeks. Everything else has some kind of problems, from instability for size to heterogeneity to preferred orientation. All of these problems will make the 3D structure determination not impossible, but really complicated. A lot of these issues, if not all, can be somewhat dealt with during the Cryo-EM experiment, from adding detergents or other additives to the sample, to carefully choosing the correct grid, to using different vitrification methods, or sample-specific data collection strategies. This is all feasible, but it translates in a much longer timeline and, of course, in a much more expensive experiment. That's why having a good sample to start with is so important. And that's why much attention should be paid to all the steps that happen before the sample even hits the grids. And it becomes more important, even more important, when the sample is not for being, let's say, less friendly, for example, multi-protein complexes. And today's webinar is about one of these complicated systems. So target protein degradation is a technology that has emerged in the past few years as a very promising approach to address specific diseases by removing specific disease-associated protein by using the cell's own destruction machinery. Different targeted protein degradation strategies have been explored, including, but not limited to, PROTACs, for example, molecular glue, lysosome targeting chimera, or antibody-based PROTACs. Proteolysis targeting chimeras, or PROTACs, were first reported by Sakamoto et al. In 2001. PROTACs are heterobifunctional molecules that contain three components: the protein of interest binding moiety, an E3 ubiquitin ligase binding moiety, and then a linker in between that joins the two head groups. What happens is that PROTACs molecule can link together the E3 ligase and the target protein, induce some proximity of the target and the ligase, so that the target can then be polyubiquitinated and subsequently subjected to proteasomal degradation. The most targeted E3 ligases by PROTACs are cereblon, CRBN, and the von Hippel-London VHL protein. And this is just because a very practical thing, that there are a lot of well-characterized small molecule binders for those two proteins that already have acceptable physicochemical properties and high binding affinities. And the problem of finding a good PROTAC is basically now boils down to only find a good protein of interest target binding molecule. Of course, a lot of structural biology projects are very well helped when we do have a structural biology component, so we have structures. And if we look through the PDB, it shows that there are about over 60 structures of complexes containing the DB1, CERAB, and heterodimer. About 2/3 of those were solved using X-rays, about 1/3 were solved using Cryo-EM, and the number of Cryo-EM structures is actually increasing as we speak. If we look a little bit more deeper in this collection of structures, we see that the CRBN DDB1 heterodimer alone, so the two components, the green slice, represents a small subset of the entire populations. About 1/4, three components, the purple slice, are composed by the CRBN DDB1 plus a specific ligand, pomalidomide or any other derivatives. The large majority of the structure are four-component structures, so they contain the DDB1, CRBN, the linker, and then a protein of interest. And this protein of interest represented in the PDB are zinc finger proteins like Icarus, but there are also several bromodomain and kinases. The bottom line of this slide is that the linker and the protein of interest are project-specific. On the other hand, the combo DDB1-CRBN is an invariable component that's common to all the structures. And so the availability of good structural quality CRBN DDB1 becomes very important to ensure success, because we already have half of the sample that we know is going to be good for structural determination. For a Cryo-EM experiment, good quality translates in a sample that is expressed in large quantities, is active, conformationally stable. And even if we can buy this protein from many, many vendors, sometimes the protein is really good for in vivo assays, but does not have the structural qualities that makes it suitable for structural determination. With this in mind, we decided to create and optimize a workflow that will allow the production of a structurally enabled DDB1 CRBN complex, and we use Cryo-EM structure determination as a final readout. And with that, I'll give the floor to Lindsey.
Yeah, so I'm going to speak a little bit about how we express and purify these targets for the work that Giovanna started to speak about already. So at the beginning of our process, we're going to look to design constructs, whether that includes various signal sequences, tags, or proteolytic sites. We're also going to assess what the most appropriate expression system is and the cell type within that expression system. In this case, we know that insect cells is a suitable expression system. And then we're also going to look, since we're using insect cells, at various conditions that can be optimized to achieve good expression, whether that be the MOI, the ratio of each virus used in the infection, or the time in culture. So once we've established all of those components, we do some optimization. In this case, we looked specifically at optimizing the MOI. So we're doing a co-infection strategy with each of the viruses, encoding the CRBN, which has a his tag at the N-terminus that's removable, and then the DDB1 subunit, which is untagged. So we do an expression screen and a single affinity step purification. Assess the samples at that point to see what our optimal MOI ratios are for each of the viruses. And we determine after we assess 3 different MOI ratios that the best case scenario for us is a 1:4 ratio of virus, that being of the CRBN DDB1. Once these expression optimization is complete, we move into purification. So as I mentioned, the CRBN component has an N-terminal his tag. So we first perform nickel NTA affinity chromatography. Subsequently, we can do a proteolytic cleavage step to remove that tag to leave us with more of a wild type sequence, and then a negative affinity chromatography step to remove any residual tagged components or proteases in the solution. And then finally, we perform a polishing step using size exclusion chromatography. For this case, we know that we're looking for the complex formation of the 2 individual subunits. So in our preparative SEC chromatography, we can see that that complex is eluting in a nice symmetrical peak. And the fraction analysis is shown below the chromatogram with the pre-SEC pool shown in lane 2, and then the subsequent fractions collected along that chromatogram shown along the remainder of the gel. So once we have gone through the purification scheme, we look to do some final analysis of the sample to assess quality. We do SDS page analysis again of that final concentrated pool under reduced and non-reduced conditions. In this case, we still see the 2 subunits present. We also do intact mass analysis, and in this case, we can identify both components being present in the sample, of which the CRBN component is at least probably more than 50% singly phosphorylated, likely at the serine 25 position. And then the DDB1 component appears to be processed to remove the N-terminal methionine and potentially a acetylation at the serine 2 position. And then finally, we're looking to assess the homogeneity of the sample. So we do analytical HPLC SEC. We can see that before a freeze-thaw cycle, we have approximately 94% intact complex in the sample. And then there is some instability we see after freeze thaw where we see a slight decrease in the amount of intact complex there, in that peak showing up at about a retention time of 8 minutes.
And I'm back. So the protein looks really good from a biochemical point of view. When we start a Cryo-EM project, we usually do a negative staining assessment to assess the quality of the sample, even if all the previous data look that they are good. But as Lindsey mentioned, there was a sign of degradation upon freeze thaw. So we wanted to see, for example, if that was going to influence the quality of the sample that we have at hand. For doing that, we use negative staining. You all know negative staining involves to depositing the sample, covering it with a heavy metal, and then taking images in the microscope. It doesn't allow you to go to high resolution, but it is much less disruptive of the sample than the process of vitrification, for example. And so it's really good to tell us how actually good the sample is in solution before we proceed to the vitrification. We can answer a question as, did the sample survive freeze thaw? And we had a question mark there. Is the sample aggregating? Is the sample monodisperse? These are all answers that can be done just by a straight micrograph. And as we can see here, in the case of the CRBN DDB1 that was produced in-house, the sample actually looks pretty good. There is no sign of aggregation, and it's fully monodisperse. The other question that we can ask using negative staining is the complex intact, or there is a dissociation at any level because of freeze thaw, because of dilution, because of anything else? In order to do that, we just collect a data set, which is about 200 images, and we do a 2D classification analysis. And what the 2D classification showed in this case is that the complex is actually formed, it is stable, and there is clearly no dissociation or not visible at this point. Both components are there, both the DDB1 and the CRBN. Even at the very low concentration, we are talking about 15 nanograms per milliliters of protein that we are using. So the biochemical characterization and the negative staining characterization clearly say that the sample is good and we can proceed to vitrification. They are also telling us that if we see problems during the vitrification, the vitrification itself is not the sample. And that is a very important thing to know before we start with a new cryo-EM experiment. So during the first round of vitrification, we do our standard screening, and we identified immediately or very quickly a condition in which the sample was not aggregated again. The particles appeared to be very monodispersed. It looked good. We did an overnight data collection, and then we calculated the 2D classes. And unfortunately, the 2D classes show only the presence of the DB1, suggesting that the sample was highly unstable under these specific vitrification conditions. We know it's not unstable because of the previous data that we had, but this vitrification condition clearly needed to be modified. The second round of vitrification, we focused then on using continuous substrate grids, like continuous carbon or single-layer graphene or graphene oxide. And we identified the graphene oxide grids as a possible alternative. They allowed for a good distribution of particles. Trust me on that. I know it's very difficult to see. And most importantly, when we calculated the 2D classes, they indicated that the sample was intact and that multiple views were also visible. So we have secondary structure elements visible. So all in all, the condition identified is suitable for high resolution structure determination. But there is always a but, unfortunately. The first data set that we collected was an extensive data set on the glaciers, and we were able to obtain a reconstruction to about 3.4 angstroms. But when we looked at the map did not look like a 3.4 angstrom map, and it was clear that there was some problem, and it was preferred orientation. And this was actually visualized fairly nicely in CryoSpark, where you can run this kind of analysis. To address the preferred orientation issue, we decided to collect a second data set using, in this case, a Cryo's microscope and tilting the stage about 30 degrees. Again, we were able to obtain structure at around 3.4 angstroms, in which the preferred orientation was, if not completely, but at least partially almost well resolved. But then, of course, when you tilt the stages, there are a lot of other problems that come in. Difference in ice thickness, increased size motion, the image motion, increase loss of signal-to-noise ratio. And that will probably account for the not as high or medium resolution that we were able to obtain even on our Cryo's microscope. Another problem that is very well known for this sample is, as I mentioned before, that is extremely flexible, which makes also achieving high resolution data quite complicated. At the end, we were able to collect with the Cryos, processing the Cryos, we did many rounds of 2D and 3D classification, and we identified two major classes that contain enough particles that warranted further 3D refinement. In both cases, the complex appears to be extremely flexible, as in fact visualized by the local resolution maps. The core of the DB1 actually is refined to better than 3 angstrom resolution, but the beta propeller domain of the DB1, as well as the CRB and moiety, appear to be at a much lower local resolution. In both cases, the CRB1 is what is called an open conformation, which is what typical of the unliganded sample. The coordinates from this PDB file, which is exactly this complex, fit fairly nicely in our electron density map, in our map. The difference between the two classes, class 1 and class 2, is just the position or the orientation of the beta propeller with respect of the entire complex. And in class 1 assumes what has been defined as a linear conformation, or conformation. In class 2 is what is called a twisted conformation. But other than that, the two classes are fairly identical. We wanted also to see if our sample was capable of binding a ligand. And the ligand we choose is iberdomide, is this one, is an experimental drug. It belongs to a class of compounds that are called cereblon E3 ligase modulators, and they are used to treat some kind of cancers. There are two structures in the PDB of the iberdomide bound to the DB1 CRBN complex, an X-ray structures and a Cryo-EM structure. The Cryo-EM structure was also solved in presence of Icarus, which is this purple magenta helix up here. The ligand in both cases is visualized as a spheres, red and purple spheres. The two structures are remarkably similar. If we eliminate the beta propeller, which is clearly in two different conformations, the two structures align with an RMSD of less than 0.6 angstroms, which is quite remarkable considering the intrinsic flexibility of this system and the fact that they were obtained with two very different techniques. As for the positioning of the ligand, the electron density of the X-ray map is clearly well defined and really support the positioning, orientation, and interactions described for the ligands, which is the golden map, the golden structure here. In the Cryo-EM structure, the resolution is much lower, and so even if the density is clearly consistent with a closed conformation of a CRBN, and so with a liganded form of the complex, there is really not enough data to support the positioning, the fully resolved ligand. In our case, the sample was, as expected, conformationally and very heterogeneous, even more than the EPO sample. One thing, for example, was that there were a lot of complexes that were representative only of the open conformation, so the unliganded species. And so we got rid of basically half of the sample just by eliminating those complexes. The remaining particles were subjected to several more rounds of 3D classifications and 3D variability analysis. And at the end, we identified three major classes, corresponding to conformational variation on both the DB1 beta propeller and the CRBN. These three classes were refined to a nominal resolution of about 4 angstroms. The first two differ mostly because of the orientation of the positioning of the beta propeller. And the CRBN is in both cases in a close conformation, consistent with the liganded form, although again, because of the medium resolution, we were not able to fully characterize the ligand. The third class appears to be either a mixture of the open and closed conformation that we were not be able to resolve, or maybe an intermediate form between the completely open and completely closed form. But again, with this resolution, we were not able to clearly differentiate between the two of them. In summary, what we have shown here is that a careful selection of parameters for expression and purification allowed us to obtain a sample that is pure, stable, and inactive. And this was confirmed by negative staining and Cryo-EM experiments, suggesting that the sample, as is, can be used to generate a quaternary complex that will be amenable to 3D reconstruction. We have also able to address, in part, the preferred orientation and flexibility issues of this binary complex, but these problems may be actually reduced when other components are present, since they can affect the particle distribution and sample orientation. Last but not least, defining that the sample is good in solution before vitrification, I think is always the safest way and time and money saving way to initiate a project. And with that, I would like to thank you very much for your attention and open the floor for questions.
I want to thank both Giovanna and Lindsey for a really, really excellent presentation. We were at the TPD conference last week, and it's clear this is a very exciting field. So we're now going to go into the live Q&A session of the webinar. So for all of the attendees, please feel free to type your questions in the Q&A toolbar, and we will address them here. If there are any questions that we do not get to, we will follow up with you by email. And just as a reminder, anybody who registered for this webinar will receive both a copy of the recording via email and also early access to our white paper on this topic. So with that, we'll get into the questions. So the first question is from Jennifer, who asked, and this is for Giovanna, can you see the different conformations of cereblon in negative stain?
We have never seen them, but we have never looked for them. So I think that it might be possible because the difference between the fully open and the fully closed, it's pretty big. And so even with the low resolution of negative staining, if we do a careful enough to declassification and with focus on that, we might be able to do it. We never looked for it. We were just looking for, is there a complex or not? And that was what we did.
Our next question is from Mike. This is again for Giovanna. So he asked, you were talking about how the complex was falling apart during vitrification initially. So he asked, do you ever use limited gentle cross-linking to help stabilize multi-component complexes that become unstable during vitrification?
We have tried, not very often, but we had cases in which we tried gentle and not so gentle cross-linking. It was never a very successful experiment. For one, and I think that's the major problem, is that you don't really know what you're cross-linking at any time. And so you might end up with a system that is extremely heterogeneous because of the chemical reaction that you're adding to the system. At least in our hands, we always end up with things that were much worse at the starting sample. What we found over the years is trying to really optimize the vitrification conditions, and very often using continuous support grids really helps in reducing the instability and the breaking up of these components.
Similar question from Reza asked about using cross-linkers specifically to tackle the flexibility of the complex.
Or do you want to tackle it? I mean, the flexibility is, especially with systems like this, it's one of the most interesting and most important part, right? Yes, you could. The question is always, what do you want to be looking at? Are you interested in the sample as a moving things and trying to get information about the biology of the sample? If you're interested in like maybe specific high resolution aspects or pieces of the complex, then yes, I would definitely use it. But if you look at these complexes as something that breathes a lot, and that's why they perform their biological activity, I will be a little bit reluctant in inducing some rigidity in there.
Our next question is from Joy, and this is a question for Lindsey. So Joy is asking about the components in the buffer during the purification process, so salt, glycerol, etc., to stabilize the complex.
Yeah, so those are all definitely things that we consider during purification for various applications. So we have to first and foremost, we want the protein to be stable, but we also have to consider the application. So for proteins destined for cryo-EM, we want to avoid high concentrations of glycerol and things that might interfere with that application specifically. When the application is more tolerant, then we do definitely want to consider the salt concentration for complexes, because maybe too high or too low may destabilize the complex formation. Glycerol is a nice additive for stabilizing against aggregation and things like that, keeping in mind that too much could interfere with the downstream application. And then other additives might also be stabilizing, whether they be excipients like sugars or reducing agents and things like that. So all very protein specific and complex specific things to consider.
All right, we have a question from Michael for Giovanna, still on the topic of this dissociating complex. So the initial conditions for vitrification caused it to dissociate, and then you found other conditions where it didn't associate. So he asked, do you have any idea, no matter how speculative, why some vitrification process causes it to dissociate and others do not?
Yeah, it's called air-water interface. It's not very speculative at all. We know that there is this issue that is related to the moment in which you thin out the sample solution, and for that short of a time it is exposed to the air, and the protein is attracted to this air-water interface, and everything can happen at that air-water interface. Preferred orientation is caused by interaction with the air-water interface. And complex dissociation and very often aggregation, they are all caused by that. So we know why it happens, or at least we know where it happens. We don't really know the physics of why it happens. We know that we need to find ways to reduce that impact of the air-water interface during vitrification.
And we have a question from Sriram. So did you try any other grids that had like a continuous film, so amorphous carbon or graphene, and what were those results if you tried them?
I will guess they were probably screened, because we usually screen them all. The initial screening is based on how the grids look, the micrograph look. And so what we pick to move on for data collection is our grid that look with good particle distribution and enough space to collect and everything else. So I would guess that with the other conditions that we tried, we did not achieve that. Either the particles were not there, or they were too diluted, or we couldn't see it. The introduction of a continuous substrate, of course, decreased the signal to noise as well. And so maybe the carbon was too thick and we didn't see anything. I can't remember, but I know we proceeded with the graphene oxide because the look of the grids was the best at that point.
All right, and a question for Lindsey. Can you touch on your decision to use an insect expression system?
Yeah, so in this case, it was pretty clear from the literature and the references and previous know-how that these particular constructs were most amenable to insect cell expression. We do evaluate both, like an SF9 cell line and a trichopillusin cell line to see which one of the two of those is optimal, as well as what I touched on in the talk about optimizing the multiplicity of infection and the ratio of the viruses. And like I said, in this case, it was more suitable. There are some constructs that are amenable to bacterial expression for this target, and we've worked in that system as well to produce other types of constructs that are part of this family of, or this class of proteins.
So we have a question from Rangarajan asking, is it normal to go low, so 0.07 mg per mil for graphene oxide grids compared to grids without substrate, 0.6 mg per mil, or is this sample specific?
It's very normal. Using continuous carbon or graphene oxide, continuous substrate grids, is like preparing grids for negative staining. They really get stuck to the grid. And so there is no protein loss to the air-water interface or on the different levels. And so normally we use concentrations that are 10 to 100-fold less than what we use for vitrification using normal holy grids.
The next question is from Yu Zhao, who asked, will you be able to support structural characterization of ternary complexes by Cryo-EM if we're able to supply compound and substrate? Giovanna.
A compound, so the protein of interest and the substrate in the PROTAC, let's say, yes, but it will be up to you to figure it out how to make the complex. So we can make it if you tell us how you want it to be made. I am not an expert on the field, but I know that there are a lot of subtleties in the ratio of the different components that you have to use in order to prepare a complex that is stable and is not dissociating too fast or not associating at all. So yeah, we can. Our protein is definitely good enough. Before moving on to the quaternary complex, we will probably need some biochemical data as well.
And Lindsey, for people who are interested in getting cereblon DDB1 from Proteos, can you touch on what you would expect for the timeline for production?
Of course. So we can produce this protein for research applications, including others, Cryo-EM or others. Our workflow would be to do a custom sort of on-demand production, especially because of the stability of this protein. Then we can know that it's freshly prepared and of the highest quality. And the turnaround time for a sort of on-demand production for this protein is about 4 weeks, depending on the lead time that we have and the communications that we have ongoing. It could be as little as 2 weeks. So if it's a need, by all means, you're welcome to contact us.
We have a lot of really excellent questions from everybody. If we do not get to your question, we will follow up with you. So I'll just end with one last question from Mohammed, which is asking, did you guys use any different ligands to test the complex formation?
No, that was the only one that we had in-house. It was the only one that we tested.
All right. Well, I know we went a little over time, but I want to thank everybody for joining us today for today's webinar. I want to give a very big thank you to Giovanna and Lindsey for the excellent presentation, for answering all of our questions. So again, you will receive an email from us with a recording of today's webinar and also the white paper detailing these studies. So if you have any additional questions or you'd like to get in touch with us to learn about either the services offered by Proteos or NIS or both, please feel free to email us at info@nanoimagingservices.com. And so with that, we will conclude today's webinar. Thank you all, and we hope to see you at the next one.
Thank you.